This is going to be another paper on the COVID vaccines, specifically mRNA-based vaccines, where in this review paper they're going to discuss the design and self-assembly of mRNA vaccines. And self-assembly is a spontaneous organization of individual molecules that allows for the design of nanoparticles with customizable properties. So for a little background, mRNA is fragile and it's very susceptible to physical, chemical, and enzymatic degradation. And this delicate nature of mRNA imposes a limitation on its storage conditions and the administration route, how it's administered to a patient. So that's the first challenge. The second challenge is that you need to precisely target an antigen that is repetitive on the disease virus. And for our mRNA vaccine, that's going to be the spike protein. The fear is that if this is not targeted appropriately, then you're not going to develop an immune response appropriately when your body actually encounters the virus. And last, naked mRNA has a limited capacity to pass physiological barriers of the body and enter the target cells, so it actually has to be delivered to the target cells. So for our mRNA, it's going to be based off of lipid nanoparticles, and we talked about how they sort of assemble around the mRNA using simple interactions as well as just high-speed mixing. So that's what we mean when we're talking about self-assembly for a delivery vessel of the mRNA. So why do mRNA over something like a live attenuated or inactivated form of the virus? Well, the first problem with doing something like this is that you can risk getting infected. This is not a risk for mRNA vaccines because you're not actually delivering live virus or a weakened virus. Second, there's the risk of endotoxins. So this is when the virus dissolves. It could release something into your body as it's breaking down that could be toxic to you. An example of this, like in a bacteria cell, could be botulism, which is essentially food poisoning that grows from improperly sterilized canned meats in general. There is also no risk of mRNA vaccines causing some sort of genomic integration of the virus into your genome, so there's no mutagenesis risk. And then there's also a high efficacy level because you're basically delivering very accurate antigenic targets for the virus you want to protect against, which develops a very robust immune response to those antigens, creating antibodies that are appropriate to help you clear the virus. Where this is also minimizing adverse effects as compared to whole cell vaccines. And then there's even manufacturing benefits. Because you're producing mRNA, it's relatively robust and scalable when compared to culture-based production of DNA or protein-based vaccines. And then, so like this next sentence is pretty important. So less optimization is required for synthesizing new antigen sequences of similar size. So this is just a fancy way of saying that for upgraded versions of the vaccine, you have to do less. And it's very, very quick and rapid. So, for example, the National Institute of Allergy and Infectious Diseases of the US and Moderna produced clinical-grade mRNA vaccines for COVID within 27 days after the sequence of the virus of the spike protein was released. It enabled the human clinical trials for phase 1, 2, and 3 to be initiated at unprecedented speed within 66 days, 140 days, and 199 days after the viral sequence was determined. So besides the clinical speed being very quick, it took 27 days from first sequence to being able to produce this. So I had a question about how does mRNA get degraded? Can it just be used forever and repeatedly? I'll cover this more in other papers that I've read on this. But for now, it's enough to know this sentence right here. So mRNA alone struggles to enter the cell because of electrostatic repulsions. So the mRNA is going to be negatively charged. And in general, there's essentially a positive charge on the outside of the cell, so it's just going to stick to the outside of the cell. It's not going to enter. Again, this is painting with a large brush. And then mRNA is prone to degradation due to abundant ribonucleases, RNases, in the extracellular space. So when it's just free-floating, there's enzymes that will quickly degrade the mRNA that's just sitting out there. And then we even have receptors in our body that mRNA will just kind of stick to. which is going to prevent it from being read and used appropriately. And that's essentially why we have to package it in these lipid nanoparticles to prevent it from interacting with enzymes, from degrading it, from preventing it from sticking to the outside of the cell, and then also to prevent it from sticking to things you don't want it to, like toll-like receptors. And early studies have showed that lipid nanoparticles enhanced the in vivo production of the targeted antigens by tenfold. It was 10 times greater than just putting the mRNA in there. When it was packaged in this delivery vehicle, this lipid shell, it had a 10 times greater production of antigens than just the mRNA alone. Now, this was one of the big revolutions that won the Nobel Prize, and it essentially comes down to this sentence right here, substituting uridine for pseudouridine. Pseudouridine is a naturally occurring modified nucleotide that is in our body. So by doing this little substitution, you're sort of tricking the body into thinking this is an mRNA template that your own body produced, which prevents any undesirable immune reactions from occurring that prevent the mRNA from being delivered to the ribosomes, which results in increased protein translation of the mRNA templates. Now, there are 3 different ways that mRNA vaccination can confer immunity to you. There's the transfection of the somatic cells, such as the muscle cells and the epidermal cells. So this would be the tissue in the immediate area where the injection was placed in your arm, so in that deltoid muscle. And then there's the transfection of the tissue-resident immune cells at the injection site. So we have the local muscle cells and skin cells, and then we also have the immune cells that are close by. But then we have a third way that this can happen, and it's sort of you have your lipid nanoparticles being grabbed and transported down to a lymph node, and then the lymph node is where it sort of produces the antibody response, and that's very, very desirable. So when you feel your lymph nodes swelling after your injection, like around your neck, that's a very good sign that you're having a good, robust immune response and that you'll have a high level of protection. Same with in your armpits, those lymph nodes there as well. And this is a fun visual of that. So you have the injection there, the local tissues can be where the immunity gets conferred, and then the local immune response. But then you can get very lucky, and some of this can transport to your lymph nodes and then also to your spleen as well. I forgot to mention that, which will help to transfer these dendritic cells into native T cells, and which can then confer immunity to active T cells and B cells. So essentially, if you're getting immune responses in all these different ways, it's a very good thing. And while this is all very fantastic, there's a couple limitations to the mRNA vaccines. The major one being that the lipid nanoparticles currently require strict storage conditions, which limits their distribution locations. So you can't distribute these to places where cold storage is scarce or unavailable. The synthesis of these products are multi-step, so it requires a lot of very specific, highly developed building blocks to be able to get to the point where they can be injected in your arm. So it's not something that a little startup company can do, unfortunately. And then there's still lots of areas where optimization can be made because this is still a very young field. But despite being young, it is a very exciting field. It's one that's based in a lot of well-established knowledge, and it is certainly a step forward in our vaccination sciences, in our immune sciences.